Hello everyone, today we're going to be going over the August 2024 Living Environment Regents Exam. In this video, we're going to be going over Part D, which is a mix of 13 short response and multiple choice questions, each worth 1 point. A link to this test can be found in the description of the video below if you want to follow along at home. That being said, we're going to start off with number 73, which says that in the relationships and biodiversity labs, the enzyme used for gel electrophoresis cut the DNA into fragments by recognizing the sequence CCGG and cutting between the C and the G. Using the same enzyme on a DNA sequence below, how many fragments would be generated? So this enzyme, its entire job is to read a long strand of DNA and just create cuts along that DNA. And it's not going to make random cuts. It's going to make cuts at specific junctions or specific markers. So in this case, this enzyme makes cuts specifically at CCGG location. So any part of this DNA that has a CCGG, the enzyme is specially designed to read this and understand that, hey, here I need to separate the C's and the G's with a line. Okay, so that's what it does. So our job here is to kind of be like that restriction enzyme and read this DNA and cut it accordingly. So if I read this DNA, I see that I have a CCGG at this location right here. So if I have a CCGG location there, I'm going to have to make a cut over here. Then I'm going to keep on reading this, and I see that I have another CCGG right over here. Okay, so now I need to make another cut over here. And if I continue reading my DNA, I see that I no longer have any more CCGGs. So I'm going to make 2 cuts, and that's going to split this DNA into 1, 2, and 3 fragments. So the correct answer choice is 3. Don't fall for the trap. They're not asking you how many cuts. They're asking you how many fragments are formed. Number 74 gives us this, is based on this big pie chart. You might be saying, well, how is this a pie chart? Well, it's just a bunch of pie charts stacked on one another. So the first pie chart shows their diets. The second pie chart shows the distribution of the types of bills. The other one shows the distribution, what percent or how many of them, the types of bills, right? Or the function of the bills. And finally, the outermost pie chart shows the distribution of the different beaks, okay? So essentially, number 74 is asking how many bird species represented in the diagram are strictly carnivores. So when we're talking about carnivores and herbivores or diet, we're going to be looking at the type of food that these animals eat. Now, when you're looking for something that's strictly carnivorous, you're looking at animals that only eat meat. Remember, carnivores are animals that eat other animals, aka meat. And when something is strictly carnivorous, that means they cannot eat anything else other than animals, right? Or animal food or meat. So in that case, there's only going to be one finch that fulfills that requirement, and that's going to be the warbler finch. If we look at the innermost chart, we see that all of these birds eat mainly plant food. We want strictly carnivores, right? So we want animals, or in this case finches, that only eat meat. So these are out of the question. Then over here we have these finches that mainly eat animal food. Well, again, remember that we're not looking for mainly, we're looking for 100% or strictly. Strictly means only or 100%. So only one of these beaks or birds is going to be 100% carnivorous, and that's going to be the warbler finch. So only one bird species is strictly carnivorous. So don't fall for the trap. Don't think that mainly animal food and 100% animal food is the same thing. It's really not, right? If something is mainly animal food, again, that means that they could be eating some plants, but strictly carnivorous means you can only eat meat. So there's only one that's 100%. 75 says different species of the Galapagos finches have beaks of unique shapes and sizes. These differences are most likely due to finches adapting to their different environments. The whole reason why any sort of species has differences within its populations is because over time they adapt to live in their environment, right? The whole reason why all of these finches have different beaks is because they live in a different part of the Galapagos Islands is a system of many islands. So some of these islands have, special tree nuts that grow on trees that require you to crush them because the nuts are very large. Some of these islands don't have any nuts, they have seeds on the ground. That means that you need to get a very long beak to kind of poke around the ground to get those seeds out. Some of the islands may not have any seeds or nuts, so you have to go for bugs like the warbler finch, right, that eats 100% animal food. So again, any sort of difference or alteration in a population is going to be due to adaptations. Why does a cheetah look different from a lion? Because those two animals have adapted to their environments, right? Cheetahs live in an environment where all of the animals are super, super quick. So the cheetah evolves to be extremely quick to chase down its prey, whereas the lion needs more strength to kill its prey. Again, you can do this example over and over and over again. The way that animals are is because they adapt to their environments. Okay, all of these other choices don't make any sense. Choice 4 wouldn't make any sense because feeding on small and large seeds. Well, one of these birds doesn't even feed on small or large seeds. They feed on other animals. And 2, just because you change locations, it doesn't matter if the birds came from South America. Just because you arrived into a location doesn't mean you're automatically going to be well adapted to that environment. Adaptation takes many generations. All right, moving on to number 76. It says, based on the sequences, which species, X, Y, or Z, is most closely related to Botanicurus? And over here, they give us an mRNA codon chart. Okay, so this gives us a hint that we're going to be looking at amino acid sequences. Otherwise, they wouldn't give it to us. And over here, they tell us that Botanicurus has this sort of DNA sequence, and then all these other species have different DNA sequences. So when we want to check for relation, or if someone's closely related to someone, we're going to see how similar their DNA is. Why? Well, DNA is this master blueprint for everything. The way you are is because of your DNA. How come? Because your DNA creates your RNA, and your RNA creates your amino acids, and your amino acids create your proteins. And the proteins inside of your body is what makes you you, right? Melanin, and melanin receptors are all protein that determine your skin color, your hair color, your eye color. There's proteins that affect how tall you grow. There's proteins that affect some diseases that you get. So again, proteins control almost every aspect of who you are. And where do these proteins come from? Well, they all originate in the DNA. So if you're identical to someone, that means you have the same proteins, the same amino acids, and if it all comes from the DNA, then you have the same DNA. So we're going to see which one of these has the most similar amount of DNA, or the most similar DNA sequence. So if we start reading from left to right, we see that they all have the first set of DNA, or the first 3 letters of their DNA sequence is the same. All of these species have CAC. Okay. Then if we look at the second row, or this second group of DNA sequence, we see that X and Y have a different DNA sequence from Botanicurus and Z. These guys have GTG, this guy has GTA, and this guy has GTA. So automatically, species Z has more in common with Botanicurus than X and Y, because they lack this part of the genetic sequence. Okay, so we continue reading. They all have GAC, so that's not special. Then over here we see that Botanicurus has TGA, while X and Y have AGA, and Z has TGA. So Z is the only species that has that same sequence, right? No one else has the same sequence there. Then we continue reading. We say that they all have GGA, so that's not special. And again, over here we see that Botanicurus has CTC, and Z has CTT, X and Y have CAC. So all of these are different. So this is one big mess. And finally, they have the same final, 3 letters in their DNA sequence. So reading this, we can see that choice Z, or species Z, is most related to Botanicurus because it shares this GTA and this TGA that no other species has. So again, species Z has to be the most similar because it has the most closely linked DNA sequence, right? It's the most similar. It has the least amount of differences. X and Y have differences here. They have differences here as well. species Z does not have those differences, right? So these have three differences: GTG, AGA, and CAC. So that's three differences, and species Z only has one difference, so it's the most closely related one. So we can eliminate choices 2 and 3, and now we're selecting between choices 1 and 4. Choice 1 says it has to be species Z because there's no DNA sequence differences. Well, that's wrong because it has one difference, so that means that the correct answer has to be 4, species Z, because there are no amino acid differences. So what that means is even though they have a difference in their DNA, the same amino acids can still be produced. And this makes sense because look at, for example, in this box of the table, right? You can have GGU, GGC, GGA, and GGG, and they'll all code, or they all correspond to that same amino acid, right? So even if you have some differences in your DNA, you can still produce the same protein, okay? So choice 4 is saying that even though they have this difference, they can still produce the same amino acid. So let's check, right? CTT turns into GAA when it's in the RNA form. And remember, this table is in the RNA form. And CTC turns into GAG when it's in the RNA form. Okay, so the way that you read this table is you look at the first base, which in both of these cases is G. Then you look at the second base, which is going to be A. So you go into the column that has A in it. So we're looking at this column. And the third base is going to be A. So we're looking at GAA, which corresponds to GLU. And GAG also corresponds to GLU. So even though they had a different DNA sequence there, they still produce the same amino acid and thus proteins. Okay, so that's why choice 4 has to be correct. The same cannot be said about species X or Y. They're different. Number 77 says, or it gives us this situation. It says that the diagram represents a model cell with a semi-permeable membrane. The contents of the model cell and beaker are labeled. So inside of the beaker is going to be water, and inside of that water is a starch indicator solution. And then inside of this plastic bag, which is a semi-permeable membrane, this means that it allows stuff to cross it. So things can cross the membrane. Okay, so if you have some molecules over here, they can pass through the membrane won't block it. Okay, so what happens is the starch indicator, any sort of indicator in a solution, is supposed to change color whenever it meets with the thing it's supposed to indicate. So starch indicator solution is supposed to change color, which is how it indicates that starch is present. Okay, and then glucose is just sugar and starch is just starch. So what happens is after you place this bag inside of water, the water now has glucose in it, and the starch indicator solution seeps into the bag and changes color. So what happened here? Well, what happened here is that the glucose managed to diffuse across this membrane, right? And you might be asking, what is diffusion? Well, that's when you go from a level of high concentration to an area of low concentration, okay? And we can do that because we have a semi-permeable membrane that will allow glucose to pass through it. So what happens in this case is that this glucose, there's a high concentration of glucose. There's a lot of this glucose inside of this bag. There is no glucose inside of the water. So some of the glucose will move outside of the bag and into the water. So that's how the glucose got here. It diffused across the membrane, okay? And the same thing happened with the water. Remember, high concentration to low concentration. So the water has high concentration here and no concentration here. Concentration is just how much is there, right? So high concentration means it's a lot there, low means there's little to no there, okay? So there's no water inside of the bag, there's a lot of water on the outside of the bag. So the water is going to diffuse inside of the bag. Remember that the water contains starch indicator solution. So not only does water get in, but the water had starch indicator solution attached to it. So this indicator flowed inside of the bag, reacted with the starch, and changed color. Okay, and the starch stayed there because it wasn't allowed to leave because of that semi-permeable membrane. Semi-permeable means it's only allowed to let certain things through, right? Semi doesn't mean all, it means some. So they're asking us one reason for the color change in the model cell predicted in the final diagram. Well, what did we just say? We just said that the water, which carries the starch indicator solution, diffused from a high concentration outside to a low concentration inside, and it reacted with the starch. So we can just say that the starch indicator solution diffused into the cell and reacted with the starch to change color. That's all that happens. So for this problem, you need to understand diffusion, or when it comes to water, it's dealing with osmosis, right? So diffusion, so nor anything can diffuse, right? Particles can diffuse, gas can diffuse, but when we're talking about water moving from high to low concentration, it's called osmosis. Okay, that just you didn't obviously that didn't need to be stated in this problem, but that's just a general difference, right? In vocabulary. So 78 is still related to this problem, and it asks us to predict how the appearance of human blood cells would change if they were placed into a concentrated salt solution. So let's just pretend that we took like one blood cell and we put it into this container of water, and the water has concentrated salt solution. So a solution is going to be a mix of a solvent, which is the liquid, and the solute, which is in this case the salt. So a solvent is the liquid, right? That's the thing that's going to do the dissolving. So if you pour salt into water, the salt obviously doesn't stay in the water, it dissolves, right? So the thing that dissolves is the solute, that's the salt, and the thing that dissolves it is the water in this case, okay? So over here we have a red blood cell, all right? And it's placed into a high concentrated salt solution. So in this case, the water is our solvent and the solute is our salt. So I have a lot of salt, 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 salt. Okay? So what's going to happen here is our blood isn't really salty, right? Our blood doesn't have a lot of salt in it. And the amount of salt that's inside of the solution is usually going to be more than the amount of salt inside of your blood cell. Now think about it this way, right? The salt can't enter your blood cell, right? Your blood cells, the membrane doesn't allow for that to happen, right? What's actually going to move is the water. And this is going to be that same property, right? Water likes to move from areas of high concentration to areas of low concentration, okay? Or it likes to go where there's more solute, okay? So whenever you're given a problem where you have solvent and solutes, the salt is going to be the solute. That's the thing that's going to dissolve in the liquid. The water always moves from low solute to high solute. Okay? Why? Because it's trying to balance out. If you have a glass that's super salty, right? Full of super salty water, the way that you can make it less salty is by diluting it, right? You add more water to it, and the more water you have, then the less salty it is, right? So essentially, that's what nature wants to do. It wants to balance it out. This blood cell isn't salty at all. Everything on the outside is salty. So nature wants water to move out of this blood cell and into the area with high solute, the stuff that has a lot of stuff, the liquid that has a lot of stuff dissolved into it, so that it can dilute it and make it less salty and more balanced, right? So in this case, the water is going to be leaving the cell. If that's confusing for you, just remember this rule. Water goes from low solute to high solute. Water always, and nature always wants to dilute things so that it's equal on one side and the other side. If I have a baggie of water, and the membrane is permeable, meaning that the water can go across the membrane, and it's placed into a solution that's extremely salty, with a lot of salt. Nature wants the amount of salt here, or the concentration of salt here, outside of the bag, to be the same as inside of the bag, right? So it's going to try its best to pump water to dilute the high concentration on the outside, aka the water is going from low solute, which is inside of the bag because there's nothing inside the bag, to high solute, which is outside of the bag. Whenever they ask you about blood cells, the blood cell will always have low solute and the environment will always have high solute if it's salty. Okay, so what's going to happen is when the water leaves the blood cell, well, the blood cells are going to shrivel, right? The blood cell is just full of water, right? So and most of your cells are full of water. That's what gives them their structure, right? The pressure that the water exerts on the cell membrane, it makes them puff up. So what happens is that water is going to leave and that blood cell is going to shrink, right? So and we're predicting the appearance. We're not predicting what's going to happen. We're just predicting how it will look. So we don't even have to say, oh, the water will leave or whatever. We just need to say that the blood cell will shrink. You can say that the water will leave the blood cell, but as long as you say that the blood cell will shrink or shrivel up or get smaller, you'll get full points. Right? So they say that we need to base our answer to 79 on the information and photographs below and our knowledge of biology. It says that genetic evidence indicates that the land and marine iguana species on the Galapagos Islands have a common ancestor that came from South America millions of years ago. There are 3 species of land iguanas and 1 species of marine iguana. The marine iguana is an excellent swimmer and the world's only seagoing lizard. It feeds on algae, seaweed, attached to underwater rocks and is widely distributed on all Galapagos rocky shorelines. The photos and informations below compare the pink land iguana and the Galapagos land iguanas. Okay, so the pink land iguana has a pink color with black bands. Okay, so I don't have pink on this, but let's just color this in purple, right? Because that's close enough to pink. Or I guess the iguana itself would be pink with black bands. So, sorry, this whole lizard would be pink, like that. It looks something like this, and then it would have black bands like this. So either way, it doesn't matter, right? It just has a funky color. So it's found only on Wolf Volcano in the northern Isabella Islands, and it feeds on cacti, shrubs, and grasses. Then we have the Galapagos land iguana, which is yellow to brown. It has a yellow to brownish body. So let's say that it's this color, right? It's yellowish and brown. It's found on 6 islands, and it feeds on cacti, seeds, grasses, and some animal sources. It's asking us, state one reason why the Galapagos pink land iguanas can coexist with the Wolf Volcano on can coexist on Wolf Volcano on northern Isabella Island. So why can the pink land iguanas coexist with the Galapagos land iguana, right? So here, this is the Galapagos land iguana, this is the pink land iguana. Why can they coexist? Well, usually animals coexist when they don't share resources, right? The whole reason why some animals can't coexist with each other is because they compete for the same resources and they fill the same niche. If 2 animals do not fill the same niche, then they can coexist with one another. Why do bumblebees and wasps, live in the same area? Well, that's just because they have different niches, right? Bumblebees go from flower to flower and pollinate, and wasps are more carnivorous and they, feed on garden pests, or they feed on aphids, or other insects in the garden. So they don't share resources because the wasps don't need the flowers while the bees do, and the bees don't eat other insects while the wasps do. So there's really no intersection in their needs. There's no intersection in where they live. There's no intersection in where they get their food from. So because they don't compete for the same resources, they can coexist in the same environment, okay? AKA they have different niches, and a niche is a role. So they're telling us that these 2 species of iguana can coexist already. They can already coexist. They already get along. Why? Well, number 1, they don't eat the same food, right? This guy feeds on cacti, shrubs, and grasses, while this guy feeds on cacti, seeds, grasses, and animal sources. So even though they can both eat cacti and grasses, if this guy's eating all the cacti and grasses, this yellow iguana can go ahead and feed on the seeds and animal sources. So they're not going to compete there. And again, they probably share the resources evenly. So your one reason can be just that, right? Either they occupy different niches, they occupy different roles. If you're occupying a different role, you're not going to compete. Or they have different resource requirements, or they have variation in their food sources, right? So I'm just going to say that the two iguanas have different resource requirements. Okay, or they have variations in their food sources, right? So again, in order for two animals to coexist, they either need to occupy different niches or they need to have different resource requirements. So number 80 says that students hypothesized that eating a candy bar a few minutes prior to exercising would increase the number of jumping jacks they could do in a given amount of time. Each participant ate one candy bar, waited 10 minutes, and then did jumping jacks for 1 minute. So these guys are like, well, if I eat a candy bar, will I be better at jumping jacks? And then they get this data. Student 1, after he ate the candy bar, did 35 jumping jacks in a minute. Student 2 did 52. Student 3 did 48. And on average, all the students did 45 jumping jacks. And it's asked that the question is really asking us, can the data in the table be used to support the hypothesis that the students proposed? Okay, so if you were given this data, can you make an assumption about this, or can you really say something about their data? And the answer is no, because how many jumping jacks were they doing before they ate this candy bar? I don't know. All I know, the only piece of data that I'm given is how many jumping jacks they did after they ate the candy bar. But in order for me to see whether the candy bar made them do more jumping jacks or less jumping jacks, I need to see what their baseline was, aka what their control group was. Okay? The whole reason why we do controlled experiments is so that we can recognize what was normal or what normally happens before the treatment. Okay, or before you alter your participants, right? So if I had another column here that said like before jumping jacks or before candy bar, and I had like 10 jumping jacks, 2 jumping jacks, 3 jumping jacks, and the average to this would be 5 jumping jacks, then I would conclude then yes, eating the candy bar does make you do more jumping jacks, because on average our average increased from 5 to 45 after we ate the candy bar. That's a giant increase. But guess what? They never collected that data beforehand, and because of that, I don't know what they were doing beforehand. I don't know whether they could do more jumping jacks before eating the candy bar or less, because that was never included. So can the data be used to support the conclusion? No, it cannot. There was no control in the experiment. We cannot tell how what did they want to find? They wanted to their ability to do jumping jacks changed, right? So again, it just has to be logical, right? If I gave you data after if I started my own test prep center, and I was like, if you come to my test prep center, I will guarantee that you will score 50% higher than what you were doing before, or like 10% higher on all of your test scores. And then I release my data, right? I'm like, oh, one of my students got a 70, the other got a 80, the other got a 90, the other got a 70. It won't show you if my test prep program is effective, because we don't know how they were doing in school before they went to my class, right? For all we know, there would be no difference, right? So again, whenever you do an experiment, you always need a control so that you understand whether or not your treatment, in this case the candy bar, was effective. Number 81 says that the 4 students were asked to explain the reason that the hypothesis was most likely correct. Their responses are shown in the table below, which row contains a correct explanation of how the body would use a candy bar. So how is the candy bar interacting with the body and making them do more or less jumping jacks? So student A said the candy bar would provide protein for increasing muscle mass to do the jumping jacks. Now, sure, that's what protein bars do, but increasing your muscle mass to do more jumping jacks is going to take months of training, right? If I eat a candy bar and then do a push-up, I'm not going to get swole, right? I'm not going to get massive muscles from that. Why? Because in order for me to gain muscle or gain muscle mass, I need to go to the gym consistently for months and then have protein supplements, right? This increasing in the muscle mass takes a lot of time. It's not going to take 10 minutes, right, from eating that candy bar, right? So again, student A has to be wrong. In order for you to gain muscle mass, that takes a long time. That's not a thing that happens in the span of 10 minutes, okay? Student B said that the candy bar would provide the raw materials required for cellular respiration in the muscles that do the jumping jacks. This makes sense, right? In order for your muscles to move, they need ATP. They need energy, okay? And how do you create ATP? Well, you undergo cellular respiration, okay? Your muscles, in order to do some sort of movement, need to absorb that ATP. And that ATP is produced from sugar plus oxygen. So a candy bar is usually sugary, okay? So if the candy bar is sugary, it's providing our body with the sugar that it needs to break down and form ATP. That's why we get sugar rushes, right? That's why little kids get hyper when you give them candy. Their body has so much sugar that it's all converted into ATP. Now they have all of this literal energy, to run around all the place without getting tired. Okay, so that's why student B is correct. Student C said the candy bar would increase the red blood cells' ability to carry more oxygen during the exercise. Again, that's false, right? That's not what a candy bar does. A candy bar is just a bunch of sugar. It's not going to increase your red blood cells' capacity to do anything. You would need more iron for that. 4, the candy bar would help the digestive system digest sugar faster during the exercise. That doesn't make sense because the more food you have, the slower you digest. The candy bar is giving your body more, or your stomach more things to actually digest, okay? So number 82 says, which 3 body systems are most directly involved in converting the energy in food to energy needed to do a activity such as jumping? Well, we were just such as jumping jack, sorry. So we were just talking about how in order to do that, we need to undergo cellular respiration. And what is the one giveaway or one body system that cellular respiration needs? It's the respiratory system, right? Cellular respiration. It literally has the respiratory system, or a synonym of the respiratory system, respiration, inside of it, right? So what that means is you need to respirate in order to do exercise, right? The whole reason why you need to breathe in oxygen is so that your cells have the oxygen to produce ATP so that it can power up the rest of your body. So any one of these choices that doesn't have respiratory should be automatically eliminated. So choice 3 is out. Everything else is respiratory. So far so good, okay? Now, how is your body going to break apart that candy bar? Well, it's not just going to magically be absorbed into your blood and then broken down into sugar. It's not just going to magically be broken down to sugar either the second it enters your mouth. It's going to have to be digested by your digestive system. So that eliminates choice 1. So anything that doesn't have respiratory or digestive should be removed. Okay? Why the digestive system? Because when the candy enters your stomach is going to use its acids to break down that candy bar into its basic components like sugars plus fat, and then your cells are going to use that sugar. That sugar is going to be transported inside of your blood cells to your muscle cells that are going to then convert that sugar into ATP, which then powers those muscle cells to move around and change shape, allowing you to go ahead and do your jumping jacks, okay? So that would choose circulatory system, and we would choose number 4, okay? Choice 4. We would eliminate choice 2, right? The reproductive system has absolutely nothing to do with your capacity to do exercise, right? If you wanted to create a child, you would use a reproductive system, but in that case, that's not exercise, right? Something else. You're not using that part of your body or that system of your body in order to just to do physical activity like jumping jacks. 83 said that students compared 4 varieties of plants using different tests. They looked at the plant's leaves and flowers and used the microscope to observe additional plant structures, paper chromatography to analyze pigments, and gel electrophoresis to compare DNA fragments. So it's asking us explain what the students were most likely trying to figure out by performing all these tests on the plants. So they're trying to compare DNA fragments, and this should bring us back to our previous problem over here where we were talking about DNA fragments. We were using DNA fragments to figure out the relationship between these three species and this one, right? What we were doing is we were using DNA to see which one was most closely related. So that's usually the only thing that we can really use DNA for, to see relations, right? Or see the relationship between two species. Why? Because DNA is that cellular blueprint. If two buildings have the same blueprint or same instructions, that means that it's going to be the same building. So that's the same thing for, plants or animals, right? If you have the same DNA as someone else, you guys are identical, which is highly unlikely, but identical twins exist and they have the same amount of DNA. So if you're comparing DNA fragments and you're also looking at plant structures, you're looking at, let's say that, this plant has, this type of leaf. Well, this plant has, two leaves that look like that. Are they relate? And this plant has the DNA sequence of like T, C, A, T, and this one has GACT. Are these going to be related? No, they're not going to be closely related, right? Because number one, they look different. Number two, they have different DNA sequences. So my point here is that you're doing all of these things to figure out which plants were most closely related. Okay, so again, analyzing DNA fragments should be a dead giveaway of that. Number 84 asks us to read the passage below. So, predicting the change in pulse rate due to physical activity. 3 students had different plans after school. Student A took several young children to the playground. Student B read science fiction at the library. Student C worked at the cash register of a fast food restaurant. Each of the students was healthy. Their heights and weights were the same. However, their pulse rates were quite different during their afternoon activities. Predict which student will have the lowest average pulse rate throughout the afternoon and support your answer. So in this case, pulse rate is going to be closely related to physical activities. If you go ahead and you run around or do bunching jumping jacks or push-ups, or you become extremely active, you're going to have a high pulse because your body wants to supply the rest of the body with more oxygen, and you're getting your blood pumping, right? The more active you are, the higher your pulse, right? Your pulse is how fast your heart is going to beat within a given amount of time. Your heart beats so that it can pump the rest of your body with blood that has oxygen in it, which helps you move around, right? You need oxygen in order to do any sort of physical activity. That's why if you run around for a very long time, you have a high pulse or high heart rate, because your heart is working extra hard to supply all of your body with muscles. So which one of these kids? So my whole point is that high activity, or people that are very active, or people that are doing something that's active, that's physically active, are going to have high pulses. So which one of these students is not doing something that's physically demanding? Well, watching several young children is physically demanding, and walking to the park is physically demanding, right? It's a physical activity. So student A is pretty active. Student C is working at a cash register. He's at work. He is taking orders. He's walking around. That's also, he's active. But student B, all he's doing is just sitting down. So whereas student A and B, their bodies need extra oxygen because they're moving around, student B's body doesn't need extra oxygen because he's just sitting down, right? His blood does not need to be, pumping as hard as someone who's standing up or someone who's moving down. So predict which student will have the lowest average pulse. Well, student B will have the lowest average pulse because they were the least active. Their muscles were less active, or not very active, and did not as much oxygen. Okay, so again, you need to support your answer, so that's my support, right? I'm saying that student B will have the lowest average pulse because they were the least active. And why does that matter? Well, if you're the least active, then your muscles require the least amount of oxygen, so your heart is not working as quickly or as hard. Okay, number 85, this is the last question on the test. It says the diagram below represents a DNA gel. So whenever you have a DNA gel or something that includes wells and has DNA going into wells, this is gel electrophoresis, okay? This is a way of identifying a relationship or a similarity between two individuals or two species using their, you guessed it, DNA, okay? So essentially the way that gel electrophoresis works is that you have this long strand of DNA from two or more people. And what you do is you cut this DNA up into fragments, some long and some short, and you place them down into these wells. And the shortest parts of the DNA segments are the ones that weigh the least amount, right? If something is short, it doesn't weigh a lot. So because something is pretty light, it's going to move at the quickest rate down the well. Whereas a longer piece of DNA or a longer fragment is going to be heavier and it's going to move slower. Okay, so essentially these fragments of DNA are going to settle into these wells at different areas, right, at different locations. And you're seeing which individuals have the most amount of fragments in common. Because if they have the most amount of fragments in common, that means that their DNA was cut in similar places. Okay, and if their DNA was cut in similar places, that means they have and their DNA fragments are the same weight and size. That means that number one, they have the same number of bases. Number two, their DNA was cut in the similar locations, which means they must have similar bases, or that their DNA must read the same. And three, they must be related because their DNA is most similar. So this question is asking which individuals are most likely the result of asexual reproduction from the same parent. And that's going to be individuals W and Z. Okay, if we look at this, if we read this chart, W has a fragment here, Z has a fragment here. W has a fragment here, Z has a fragment there. W has a fragment here, 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 and here. W and X do not share those specific fragments. As a matter of fact, W and Z have the same fragments in the same position. Asexual reproduction produces identical offspring. That means that they have the same DNA. So if you cut the same piece of DNA using the same enzyme, it's going to make the cuts in the exact same location, and those cuts are going to have the same amount of DNA, so it's going to weigh the same, and it's going to be the same length. So individuals W and Z are the result of asexual reproduction because the pattern of lines for W and Z are identical. Okay, so that's how you get full points on that final question. That's all 13 questions on part D of the biology regents or living environment regents gone over in one video. If you have any questions, please feel free to let me know in the comments below, or ask them in the comment below. If you have any suggestions for future videos, or you want me to go over a different part of this test, please let me know in the comments below as well. That being said, I hope you guys learned something, and I hope you guys have a nice day.